Oh no, it's me, Mycobacterium tuberculosis, but you can call me MTB for short. I'm here to tell you my tale of survival and persistence in this vast world. I may be small, but don't let my size fool you. I've been around for millions of years, adapting and evolving to thrive in almost every corner of the globe, causing devastation and suffering wherever I go. I am a ruthless, gram-positive, rod-shaped bacillus that infects nearly one-third of the world's population. Pretty scary, huh? I take pride in my title as the king of killers, given my terrifyingly high fatality rate and resistance to many antibiotics and disinfectants. According to the World Health Organization, I take the lives of 1.5 million people each year, making me the world's top infectious killer. This is all thanks to my distinctive cell wall that makes me difficult to detect and even harder to eliminate once I've taken hold. Beware, for if you cross my path, you may not live to tell the tale. My unique hydrophobic cell wall is pretty spectacular, making me a standout amongst other bacterias. It has a high lipid content, including layers of lipopolysaccharides, a layer of arabinogalactan, a polymer of arabinose and galactose, a thick layer of peptidoglycan, and mycolic acids, which give me my distinct waxy look. Mycolic acids, the long-chain fatty acids on my outer membrane, contribute to my impermeability, resistance to oxidative stress, and dehydration. They prevent the entry of small molecules, including antibiotics, and protect me from the host's immune response. My outer layer also contains various proteins, lipids, and glycolipids that play a role in my interactions with the host's immune system. All these components contribute to my ability to evade the host's immune responses, survive within the host, and cause persistent infections. I belong to the phylum Actinobacteria and the family Mycobacteriaceae, making me closely related to other mycobacteria such as Mycobacterium leprae and Mycobacterium bovis. We share similarities in our cell wall composition and metabolism, but each of us has adapted to different host environments and caused distinctly different diseases. Unlike my relatives, humans are my only reservoir. I am basically the paradigm for host pathogen adaptation, as my ancestors have paved the way for co-evolving with humans throughout history. Speaking of humans, by my beloved hosts, so fragile, so susceptible to my charms. When I infect them, I can cause anything from mild discomfort to full-blown disease. Tuberculosis, anyone? That's right, I'm one responsible for this formidable disease. The coughing, the chest pains, the fever, the weight loss, it's all part of my grand plan to spread my influence and multiply my kind. Evolving my virulence mechanisms, my lifestyle is one of constant movement and adaptation in order to complete my mission, survive, persist, and spread my colonies. I'm one scary intracellular bacterium. I am transmitted through the inhalation of droplet nuclei in the air when an infected person coughs, sneezes, or speaks, and I can travel long distances before settling down in a new host. In the depths of the lungs is where I found my true calling, to take root and flourish, feeding off rich supply of nutrients provided by my unsuspecting host. I'm what you would call a scavenger. This dark, damp, and warm oxygen-rich environment is the perfect place since I'm highly aerobic and require high levels of oxygen to survive. As soon as I enter the alveoli section of your lungs, I am encountered with a variety of other microorganisms, including alveolar macrophages. These cells are like the superheroes of the immune system, the first line of defense for the body. These macrophages quickly realize I'm an invader and phagocytize me by packing me up into a space called a phagosome. The macrophage, hard at work, tries to fuse the phagosome with the lysosome, which has a hydrolytic enzyme that can pretty much break down and destroy any bacteria. Me, being so cunning, I quickly learned how to inhibit this fusion by producing and secreting an enzyme, a lysosomotropic lipid, and a protein called tyrosine phosphatase. This allows me to evade the host's immune response and establish a localized infection. Boom, latent tuberculosis is born where I can lie dormant for years. From there, I don't just manage to survive, I begin to multiply within the macrophages, using them as a safe haven to avoid being eradicated by the immune system. As I begin to multiply and build my army and thrive within the host, I trigger an innate immune response that leads to the formation of granulomas, organized multicellular structures made up of immune cells, such as dendritic cells, T cells, B cells, neutrophils, and fibrous tissue. Once again, I cunningly manipulated the formation of these granulomas to create an environment conducive to my own survival. Within these granulomas, I continue to multiply, leading to the formation of caseous necrosis, a cheese-like mass of dead cells and bacteria. More areas of caseous necrosis are formed and begin to cavitate, allowing me to spread through airways and lymphatic channels to other parts of the body, producing serious damage to other organs and ultimately my mass domination. Another reason why I'm so successful and such a strong pathogen lies in my capacity to scavenge and adapt to the nutritional resources available to me within the host tissues. I am your typical heterotroph, fueling myself with various nutrients, especially lipids from my host and surroundings, as my primary carbon source. I also must employ various metabolic pathways to generate energy and essential cellular building blocks, primarily utilizing tricarboxylic acid cycle as well as the glycoxylate shunt to produce energy and intermediates for biosynthesis. I utilize fatty acids as an energy source through bioxidation, a process that catabolizes long-chain fatty acids into acetyl-CoA units that can be further metabolized for energy generation. This versatility in nutrient utilization is crucial for me to adapt to the diverse and constantly changing environments I encounter in the host, making me small but oh so mighty. In response to the varying harsh host environments such as varying acidity, osmolarity, and nutrient-deficient microenvironments, I switch between replicative and non-replicative states by rerouting my metabolic networks. During unfavorable conditions such as nutrient limitation or low oxygen, I lie dormant in a non-replicative state, making me viable but exhibit low or minimal metabolic activity. I can remain dormant for extended periods of time, living out my days within you in a state of hibernation or inactive. You won't even know I am there since most of you are asymptomatic. Another reason why I am so scary. For those humans who are sick, your weakened immune system wakes me up and I come back alive, inflicting pain and suffering. What you call active TB, cough, cough.
A parasite lurks in your kid's sandbox and hides in your garden beds. It contaminates your food and your water sources, and now it's patiently waiting for you. That once you meet, you will be together forever. However, it is a parasite, and you may not even be aware that you are being infected. With that being said, once it is in, it doesn't come out. It is estimated that 30% of humans worldwide are infected with toxoplasmosis. Toxoplasmosis is a global issue as it is highly infectious yet underreported. The parasite was given its name in 1908 after it was first discovered in a rodent-like animal. The parasite was later renamed after its morphology, combining the prefix toxo with the combining form plasma and the suffix gondii, together giving rise to the name used today, Toxoplasma gondii. The gondii is a zoonotic parasite, meaning it can infect any warm-blooded animal in the world. These infected animals are known as intermediate hosts. Felines are the only known definitive host in toxoplasmosis because they are the only known hosts to lack the expression of the D6D gene. This genetic mutation naturally promotes higher levels of linoleic acid as a side effect. Definitive hosts are essential to the parasite's sexual reproduction because of their metabolic processes. The parasite life cycle includes the environmental stage, the fast-replicating tachyzoite, and the slow bradiozoite. The sporozoite lives dormantly inside the oocysts that are deposited into the environment by the definitive host. The sporozoite hides safely inside its enclosed casing, which helps protect it from the environment. Once ingested, the casing begins to fall away, and the parasite begins to change, making its way to the intestine. The second stage is referred to as the fast replicating stage. It is also known as the invasive tachyzoite stage in chronic stage toxoplasmosis. Upon changing forms, tachyzoite initiates several replication cycles, also known as budding in the intermediate host. This fast replication process usually happens within the small intestine and usually only during the initial infection. This localized infection causes inflammation and symptoms of fever, lethargy, stomach aches, and general unwell feeling. Additionally, the repeated replication causes the cells to lyse, allowing the bradiozoites to travel freely through the host's bloodstream. Here they are irregularly deposited throughout the central nervous system the eyes, and the cardiac tissue. According to the CDC, toxoplasmosis is considered to be the leading cause of death attributed to foodborne illness in the United States. Accidental ingestion of infected oocysts from undercooked meat such as pork, lamb, and all backyard chickens living among domestic felines are considered more prevalent sources. Eating unwashed fruits and vegetables can also put you at risk. It is recommended to keep lids on the kids' sandboxes, wearing gloves and using great caution when cleaning litter boxes or avoiding it altogether if pregnant. Infection while pregnant is known as congenital transmission, and it carries heavy consequences for the fetus, including blindness, deafness, hydrocephalus, mental retardation, premature death, and stillbirth. If at all possible, cats should be strictly indoor pets and avoid any temptation of ingesting the infected prey. Chronic phase of infection is characterized by the reactivation of the latent bradyzoite cyst. When the bradyzoite cyst is triggered, it changes into the invasive tachyzoite, causing a tremendous amount of tissue damage. Encephalitis is a symptom that can occur when inflammation in the brain builds due to this stage of reactivation. Ocular damage is often seen, along with the presence of eye lesions and eye pain. Additionally, myocarditis, inflammation around the heart, and lung infections like pneumonia are also seen. The fecal oral route is most commonly seen in humans. Knowing that the fecal oral route is most commonly seen in humans provides us with the information we need to minimize and avoid exposure. We should remember that toxoplasmosis is typically asymptomatic if healthy. However, the disease can be destructive and even fatal, as some may not recover from the successive rounds of tachyzoite invasion. I want to draw your attention to an earlier quote. Toxoplasma gondii recognizes its final host based on the abundance of linoleic acid. Some research has been done on mice and indicates that the parasite may manipulate the behavior of the intermediate host, changing its behavior in order to find its way back to the definitive host. This innate behavior is what essentially drives the parasite life cycle. I think more awareness should be directed to this parasite and the disease it causes. Additionally, studies should be done investigating the neural behavioral manipulation specifically in the parasite-host relationship in relation to the parasite locating its definitive host, completing its life cycle. Thank you for listening.
Hello, my name is Gareth Fielking, and this is my term project presentation on methanogenic archaea and the profound effect they have on our world now and in the future. Microorganisms help shape almost every aspect of the world around us, from disease processes to processing the decay of fallen leaves, manipulation of microbes for food and production, to medical innovations. They have a profound effect and role in propping up modern society. However, no group of microorganisms is poised to have such a large effect on the globe and human population as methanogens. Methanogenic archaea that exist in the rumen of beef cattle and ovine cattle generate a high amount of methane that affects the atmospheric composition. In the future, methanogens that have been locked up in permafrost and marshlands may release enough methane to cause a tipping point event in the balance of our global climate. Methanogens are a family made up of almost exclusively archaea that are found in soil, lake beds, and deep oceanic vents. Most methanogenic archaea are from the family Methanobacteriaceae. This family includes Methanobacterium, Methanosarcina, Methanococcus, and Methanothermobacter. This last entry includes a variety of archaea that exclusively exist in deep sea hydrothermal vents and may have been some of the earliest prokaryotes to evolve on Earth. Methanogenic archaea can exist in almost as wide a variety of shapes and configurations as their bacterial counterparts, including Strepto and Staphylococcus. They can also exist in Streptobacillus configurations, which is the primary way they exist in the rumen of cattle. Methanogenic archaea's major difference is that they lack a peptidoglycan cell wall. Other substances exist in peptidoglycan's place. Most methanobacterial genera utilize a partial reductive citric acid cycle. During their cellular respiration, they primarily generate methane as a product of the fermentation of acetate, specifically activated acetate in the form of acetyl-CoA. This pathway is largely used in archaea that are strict anaerobes. These bacteria can synthesize a compound called carbon monoxide dehydrogenase, or CODH, which catalyzes the cleavage of acetyl-CoA. The resulting methyl group is eventually reduced, partially producing methane and carbon dioxide as byproducts. This metabolic loop is enhanced when the methanogenic archaea are in a system with other bacteria that are acetogenic, such as Clostridium. In this way, there is much more acetate around to ferment, which is the exact environment that is present in ruminant animals. Livestock and manure from that livestock account for 5.8% of global carbon equivalent output from human sources. I include this as a human source because it is due to the high global demand for beef and ovine products that necessitates the existence of such a large population of large ruminants. This output is driven by the methanogenic archaea not just in the rumen of the ruminant animals, but also in those that live in the decomposing waste ponds that border large cattle operations. Biowaste consumption, after all, and its disposal is one of the industrial and agricultural applications of methanogenic archaea. In fact, the top four agricultural sources of methane come from, in this order, beef, lamb and mutton, cheese production, and dairy herds. The majority of methane in this process is produced during the farming phase of production, meaning it is solely a byproduct of the methanogens at work in the rumen of the cattle. Fortunately, there are some mitigation strategies that can be employed to help with the production of methane from ruminant cattle. These methods include vaccination of cattle to limit methanogenic archaea, or introducing more dietary fatty acids to their diet, both of which target the archaea directly to reduce their methane output. Of course, the best way to reduce the impact of methane from this animal food system is to reduce our consumption of meat. It isn't a large percentage of the total carbon equivalent output, but it is significant and it is low-hanging fruit in the fight against global climate change. Thank you for listening, and I hope you found this presentation at least marginally informative.
Cheese making, an art steeped in tradition and innovation. Within this intricate world, a tiny hero plays a pivotal role, Penicillium roqueforti. This is Penicillium roqueforti, a microscopic fungus renowned for its distinctive blue-green hue. In the cheese-making process, this filamentous fungus is an architect, shaping the flavors and textures of beloved blue cheeses like Roquefort, Gorgonzola, and Stilton. Under the microscope, Penicillium roqueforti reveals its intricate anatomy, boasting branching hyphae that weave a mesh-like mycelium within the cheese. These filaments create pockets where air is trapped, forming the iconic blue veins. Phylogenetically, Penicillium roqueforti belongs to the Penicillium genus. Its genetic makeup shares ties with relatives like Penicillium chrysogenum and Penicillium camemberti. This tiny fungus is able to grow under harsh conditions such as low temperatures, low oxygen levels, high carbon dioxide concentration, and/or its resistance to organic acids and weak acid preservatives. P. roqueforti feeds on the proteins and fats present in the cheese, breaking them down into simpler compounds through enzymic action. In result, this process contributes to the unique flavors and textures seen in blue cheeses. During cheese production, Penicillium roqueforti interacts with lactic acid bacteria, creating an acidic environment that fosters its growth. This interplay is essential for crafting the distinct flavor profile of blue cheeses. Temperature, humidity, and airflow significantly influence the growth of Penicillium roqueforti. Cheesemakers manipulate these factors to control its growth and the development of the cheese. The symbiotic relationship between Penicillium roqueforti and cheese curd results in the formation of exquisite blue cheeses. However, if not controlled, certain strains can lead to spoilage in food items. In conclusion, Penicillium roqueforti stands as a microscopic architect, intricately crafting the delectable flavors and textures of beloved blue cheeses, while also holding potential in broader fields such as medicine.
Cryptococcus gattii is a fungus that was originally thought to only be in tropical and subtropical climates. It has been found now all across the globe in various areas. It's believed that global warming has allowed it to activate. This comes from the group Beardsley and their work from 2022. It lives in soil, tree bark, flowers, and various types of trees. Here in the Pacific Northwest, it is found in Douglas firs and soil. It belongs to the genus Cryptococcus, containing about 322 species. Cryptococcus gattii was thought to be a variety of Cryptococcus neoformans, but with molecular typing advancements, it has found that they have two different genotypes, and even within these two, they have a variety of subspecies. They are known as complexes. Both of them can be found in the environment as yeast cells and spores. Cryptococcus neoformans is found in pigeon guano, as well as trees and flowers. Cryptococcus gattii is also found in pigeon guano, but it doesn't seemingly reproduce well in this type of environment. They reproduce by budding or sexually with their own species or each other, creating hybrids of Cryptococcus gattii and Cryptococcus neoformans. They get their nutrients by absorbing them from the environment around them. So they need other microbes to break down macromolecules so they are more absorbable for them, for their metabolic requirements. This we know from the group of Watkins and their research in 2017. Some of the carbohydrates that they use for metabolic needs are glucose, acyclic sugar alcohols, and trehalose. Cryptococcus gattii also uses catecholamine precursors to make melanin, which is an important part of its virulence. It needs zinc for growth, for its enzymes, and as a cofactor for proteins like carbonic anhydrase. It uses lipids for energy, maintenance of its structures like the plasma membrane and intracellular membranes. Cryptococcus gattii is made up of a nucleus, mitochondria, intracellular phospholipid, bilayer membrane, a cell wall, and it can have an outer capsule or not. The capsule is made up of polysaccharides, and these components are actually made inside the cell and then deposited outside of it by exocytosis. This we know from the work of Watkins and their work done in 2017. The thickness of the capsule can vary. Outside in the environment, it is small like this, and once it gets inside a host, it enlarges, like seen here. And this is extremely important for its virulence. This sheer size keeps it from being phagocytized by the host's immune system. Melanin is another important virulence factor. It protects it from being recognized by the host immune system. It also changes its electrical charge, so it makes it less likely to be phagocytized as well. Cryptococcosis is the sickness that you get from inhaling these spores as they are out and about in the environment. In the lungs, it can cause symptoms of shortness of breath, fever, chest pain, or cough. In the central nervous system, it also causes fever, headaches, light sensitivity, seizures, confusion, or neck pain. There are various tests, both imaging tests as well as blood work tests, that can be done to see if you have a cryptococcus type disease, and then from there it can be speciated further into either neoformans or gattii, or one of the other ones that are pathogenic. So here in the Pacific Northwest, it can be found commonly in places where we go hiking with our pets, where there has been lumber that has recently been cut down, or just soil that has been disturbed, or just out and about. More research really needs to be done, as it's been fairly recently that they've separated this from Cryptococcus neoformans. So it is out as part of our normal fungi that we are exposed to on a day-to-day basis. Hopefully, as science furthers, we can find out more research that is specific to Cryptococcus gattii. For now, it's something that is out there and is unavoidable. However, it does seem to be kind of low as far as its disease process goes. And this is my presentation on Cryptococcus gattii.
Hi, welcome to my presentation on Saccharomyces cerevisiae, otherwise known as baker's yeast. People worldwide gather together to share food and drink. Bread is broken and beer is poured. The bread is light and fluffy, and the beer leaves a heady feel after a glass or two. Good times are had by all, but this would not be possible without the work of a group of minuscule fungal cells, Saccharomyces cerevisiae. Yeast has been an integral part in our baking and in our drinking since ancient times. Historians have found that the earliest known record of yeast was to help bread rise, was used in ancient Egypt in 1300 to 1500 BCE, and in China in 500 to 300 BC. The use of yeast in fermentation is much older, however. Studies show that the specific S. cerevisiae came from the east to west via China through what would be known as the Silk Road. Louis Pasteur is credited with discovering the role of yeast in fermentation in 1857. His experiments showed that fermentation and yeast multiplication occurs in parallel. He also noticed that the yeast must be alive for alcohol to be produced. Later, in 1859, scientist J. H. van der Broek visually discovered the morphology of the yeast organism. Prior to the 1700s, yeast was kept alive and sold and traded as an active culture commonly scooped up from the dregs of beer brewing. It was more commonly used for brewing than for baking. Approximately today, 1 million tons of S. cerevisiae are produced annually in the European yeast industry. Around 30% of this is exported globally. Strains of S. cerevisiae dominate the commercial use of bread yeasts, which are manufactured by Fleischmann's, Red Star, and SAF. S. cerevisiae is also present in the majority of sampled starters of sourdough. Now let's take a deeper look at S. cerevisiae. S. cerevisiae is a fungus that is classified in the family of Saccharomyces, which includes many species of yeast. It belongs in the phylum of Ascomycetes. As a eukaryote, it has chromosomes found in a nucleus and uses mitochondria to conduct cellular respiration. Its closest genetic relative is the opportunistic human fungal pathogen Candida glabrata. Its shape depends on what life cycle it is in. It can be seen as spherical singlets, budded doublets, and clusters. Its cell wall is comprised of a beta-glucan network and a small amount of chitin associated with a manoprotein layer. In 1996, it was the first fully sequenced eukaryotic genome. S. cerevisiae exists as both a haploid and a diploid cell. As a haploid, it follows a simple cycle of cell growth followed by mitosis. Under stressful conditions, these cells will die. As a diploid, the cell also undergoes a cycle of growth and mitosis. However, under stressful conditions, the cell undergoes meiosis and will produce haploid spores. These spores can then mate, producing diploid cells. S. cerevisiae is a heterotroph and obtains its energy through fermenting glucose. To do so, it utilizes both aerobic and anaerobic respiration. Yeast can ferment a large spectrum of sugars, including glucose, fructose, sucrose, maltose, and maltodextrose predominate. During fermentation, it produces ethanol and carbon dioxide. And on this slide, it has a nice little diagram of alcoholic fermentation where it shows the yeast that converts the glucose into the ADP and the ATP and the NAD plus and the NADH, into the pyruvate, into the CO2 that's used for bread baking, all the way to the ethanol that's used for alcohol. So as a result of ethanol excretion during its fermentation, S. cerevisiae kills off its competition for resources. But in a nitrogen-rich environment, it can adjust its metabolism by secreting amino acids, which enables the survival of other microbes such as Lactobacillus plantrum and Lactococcus lactic. One study grew S. cerevisiae along with L. lactis and L. plantrum. The study showed that the yeast varied little from culture to culture, but the other two organisms could only grow when co-cultured with yeast, which suggested a metabolic dependency. So aside from fermenting glucose for energy, S. cerevisiae has simple nutritional requirements. It obtains its carbon through other carbon-containing compounds, some as simple as a 2-carbon acetate. It also requires a source of nitrogen, which can be as simple as a single nitrogen-containing ammonium sulfate. And it also uses urea and various amino acids, the vitamin biotin, and a variety of salts and trace elements. So to produce wine, yeast is added to grape juice. The yeast then converts the natural sugars of the grapes into ethanol and carbon dioxide. S. cerevisiae is able to withstand the harsh environments and stressors of wine fermentation, including anaerobic conditions, depletion of nutrient reserves, increased acid concentration, ethanol toxicity, and temperature variations, thus giving it the term the wine yeast. S. cerevisiae is also the most commonly used yeast species in bread and sourdoughs, earning it the title baker's yeast. Yeast releases enzymes that break down the starch in flour to convert it to sugar. It then ferments the sugar to create ethanol and CO2. The release of CO2 is what makes the dough rise, and when heated, the ethanol produced by the yeast then converts to a gas, causing the dough to rise further. And that concludes my presentation on S. cerevisiae. I hope you guys enjoyed it. Thank you so much for listening.
Hello, I'm Gigi Coughlin, and I'm going to talk about Batrachicordytrium dendrobatitis. The common name is trithrid fungus, and it's an infamous fungus, but many researchers refer to it as BD. This fungus is infamous because of a disease it causes in amphibians called cytridiomycosis. The fungus itself is a rough spherical shape, smooth-walled, asexual, eukaryotic, and it's most infamously known for being a fungal pathogen. It belongs to the fungi kingdom and to the Batrachotitraceae family. The fungus is unique as it is aquatic and produces flagellate zoospores. A zoospore is a spore that is able to actually swim. The life cycle of BD comes in two stages. First, we can observe the zoospores dispersal, and the second lifestyle stage includes the development of monocentric thallus into a single zoosporangia. The zoosporangia produce, contain, and discharge the zoospores. The zoospores have an ovidual body shape, membrane-bound ribosomes, and most notably, they have a single flagellum. Zoospores have the single flagellum that helps the spores actually swim through water. The main way that the fungus infects a host is through the zoospores in water, but other ways are possible, like amphibian-to-amphibian contact. It's theorized that the Korean War actually began the spread of fungus, but other people have theorized that the fungus also spread through the illegal pet trade. However it did spread to the rest of the world, BD has now become infamous for causes of cytridiomycosis in amphibians. This is a skin disease that comes from the zoospores of BD. BD has now affected over 8,000 species, and it can be found in almost every continent except Antarctica. Probably not Antarctica because there's no known living species of amphibians in Antarctica. Amphibians have now become the most threatened vertebrate class in the planet. BD has caused many species to even go extinct. Species like Elatus obstetricans, known as the midwife toad, from the French Pyrenees, die quickly from the infection. But on the other hand, species like the American bulldog are actually resistant, but they can still be carriers, making them very dangerous to other amphibians. That's what I would call a real bully frog. Cytridiomycosis is spread through contaminated water because frogs drink and breathe through their skin. The fungus is able to make its way by infecting the keratinized epidermal cells of amphibians' skin and cause disease of chytridiomycosis. Scientists have found that BD unspoils the amphibians' proteins in their skin and breaks down the proteins, which results in kind of a spaghetti of amino acids. When a frog is infected, you can tell because they'll get something called red leg, and they'll be extremely sleepy and lethargic. But other than that, there's no real signs. And just a few weeks later, frogs can experience heart failure and pass away, leaving behind a mass graveyard of amphibians. Scientists have yet to find a cure to the fungal infection or a way to make environments safe from the spores. But they can apply a topical fungicide to individual frogs, which has been successful, but it still remains difficult for researchers to apply this technique to an entire population. One way scientists have helped is by breeding captive amphibians of the populations that are most affected by BD in order to keep the species alive. Mapping this fungus is extremely helpful for scientists to know the locations of BD to help minimize its spread to other areas, which is why apps like iNaturalist can be really helpful if anyone wants to help and become a citizen naturalist. Future research is also required to help minimize BD so we can protect the biodiversity of amphibians. Researchers have found that there's a host of immunity in some species where they produce antimicrobial peptides to combat the disease. This knowledge may find itself very helpful in future research when looking for a treatment. An important fact to remember is that while Chytridiomycosis from BD zoospores continues to kill many amphibians, it is still not the greatest threat to these species. The greatest threat continues to be habitat loss due to deforestation and climate change. Thank you.
Hello everyone and welcome to my term project about Schistosoma haematobium. Today we'll be going through a brief introduction followed by shape and anatomy, phylogeny, relevant relatives, microbial nutrition, growth and life cycle and the interactions and impacts of this parasite. So, this is Schistosoma haematobium in the flesh. It is a parasitic flatworm categorized as a digenetic trematode, more commonly known as a blood fluke. It primarily affects the urinary tract in humans and is the primary cause of schistosomiasis in adults, which currently affects 200 million people across 74 countries. As you can see on this slide, this is a map showing the global distribution of S. haematobium, of which the majority of cases are endemic to Africa. Approximately 85% of worldwide cases are reported in Africa, and in some local populations, prevalence rates can exceed 50%. Of note, S. haematobium can also be found in the Middle East, although it is not nearly as prevalent. Here is a representation of the growth and life cycle of S. haematobium from the CDC. It starts when eggs are shed from an infected human via urine/feces into a water source. The eggs then hatch and release myricygia. Myricygia penetrates snail tissues. Once the snails are infested, spirocysts develop. These spirocysts then are released from the snails into the water as free-swimming cercariae, which are capable of penetrating human skin. Once they have penetrated the host, the cercariae lose their tails and transform into schistosomula, which enter the bloodstream and migrate to the liver, where they mature into adults. Paired mature adult worms then migrate either to the mesenteric venules of the bowel, wherein eggs are shed in the human stool, or to the venous plexus of the bladder, where eggs are shed through the urine. As for transmission, S. haematobium cannot be transmitted from person to person. It is transmitted when infected individuals contaminate freshwater sources with urine or feces which contain the parasitic eggs that later hatch in water and infect humans bathing or working in the water. Adult worms measure about 1 to 2 centimeters in length with a cylindrical body that features 2 terminal suckers, a complex tegument, a blind digestive tract and reproductive organs. Males have a gynecophoric canal in which the female worm lives. Females are longer and thinner than males and have a uterus, ovaries and vitelline tissue. Here is a histological representation of the anatomy of an adult worm, which features the female worm nestled within the gynecophoric canal of the male worm. S. haematobium belongs to the family Schistosomatidae, with its closer relatives being Schistosoma mansoni, Schistosoma japonicum, Schistosoma mccongi and Schistosoma intercalatum. These are all blood flukes as well, with very similar modes of transmission and infection, although the areas they affect in the body differ. Here is the phylogeny of S. haematobium for comparison. As you can see, it is identical to its close relatives on the previous slide. Adult worms nourish themselves by continuously bathing in blood within the body, taking up nutrients directly across their body surface and via ingestion of blood into their gut. Male worms ingest about 100 ml per day, which is a considerable amount, while females ingest 900 ml of blood per day, which exceeds 4 times their own body volume. After ingestion, erythrocytes, red blood cells, are lysed as they pass through the specialized esophagus, while leukocytes become tethered and disabled. As mentioned earlier, S. haematobium is among the leading causes of schistosomiasis along with its relatives. Symptoms of schistosomiasis include fever, cough, abdominal pain, diarrhea, hepatosplenomegaly and eosinophilia. Infection can also cause hematuria, blood in the urine, scarring/calcification of the urinary tract and squamous cell carcinoma. Additionally, in rare cases, embolic egg granulomas form within the brain and spinal cord. Recent studies have also shown that infection with S. haematobium may be a predisposing factor for the acquisition of human immunodeficiency virus, HIV. Cancer and chronic kidney disease may also be associated with S. haematobium infection. S. haematobium has been shown to interact with other microorganisms, such as Salmonella. Schistosoma salmonella infections can occur when enteroinvasive Salmonella enter the circulation system and are able to attach to the tegument of adult Schistosoma. The schistosoma provides a refuge wherein the Salmonella bacteria can evade attempts of eradication with systemic antibiotics. While Schistosoma haematobium is proliferative within poor communities primarily in Africa with over 200 million people affected, the World Health Organization is taking strides to prevent and control the spread of schistomiasis. They are implementing improved access to safe water, improving sanitation and pushing hygiene education in affected areas. The World Health Organization is focused on reducing the disease through periodic targeted treatment with praziquantel through the large-scale treatment of affected populations, primarily targeting school-aged children, adults who are at high risk, such as fishermen, farmers and irrigation workers, and entire communities living in highly endemic areas. Thank you for listening to my presentation. I hope you have enjoyed it.
Plasmodium. Plasmodium is a parasite, a type of eukaryotic single organism of the genus Protozoa, which is transmitted by the bite of an infected female Anopheles mosquito, causing a serious life-threatening disease called malaria. Malaria is especially prevalent on tropical and subtropical areas. Plasmodium has an interesting complex structure and anatomy. It has an elongated shape, usually between 2 to 2.5 micrometers in length and about 1 micrometer in width. It is composed of two cellular membranes, an outer cell membrane called plasmalemma and an inner cytoplasmic membrane called glycocalyx. The plasmodium also possesses several unique organelles, including Golgi apparatus, endoplasmic reticulum, and mitochondria, and a specialized organ called apicoplast, which is essential for the synthesis of proteins and lipids needed for the parasite survival. Plasmodium belongs to the phylum Apicomplexa, which includes organisms such as Cryptosporidium, Toxoplasma, and Cyclospora. The closest relatives to Plasmodium are Cryptosporidium parvum and Toxoplasma gondii, both of which belong to the same genus and are closely related in terms of genetic makeup and life cycles. The Plasmodium itself is further subdivided into several species, including Plasmodium vivax, falciparum, malaria, and ovale. Each of these species is capable of causing different types of malaria in human host. Plasmodium require specific nutrients for growth and metabolism. Sources of nutrients include proteins, carbohydrates, lipids, vitamins, and minerals. The Plasmodium parasite obtain proteins from their host red blood cells and vitamin and minerals from the host plasmaserum. The metabolism and growth is highly dependable on the environment and the availability of nutrients. This parasite uses glucose as its main source of energy to generate ATP molecules that are used in the growth, replication, and metabolic processes. It's also capable of performing both aerobic and anaerobic respiration depending on the availability of oxygen. In terms of growth, Plasmodium is able to undergo asexual reproduction through a process known as schizogony, in which the parasite replicates a genetically identical daughter cell that is released into the host bloodstream. In addition, Plasmodium is also capable of undergoing sexual reproduction through a process known as gametogony. In this process, the parasite produces gametocytes that will eventually undergo meiosis to form haploid gametes that can fuse to form a zygote. The life cycle of the malaria parasite is complex. Plasmodium spends part of its life cycle in a human host and the other part in a mosquito. In the human host, the Plasmodium life cycle begins when an infected female Anopheles mosquito injects the parasite into a human's skin during a blood meal. The parasite then moves through the bloodstream, where it invades liver cells. This is the exoerythrocytic phase. Once inside the liver cells, the parasite multiplies to form thousands of daughter cells known as merocytes. Once the daughter cells are mature, they burst out of the liver and enter the bloodstream. This is where the erythrocytic phase starts. Here, the parasite invades the red blood cells, multiplies, and bursts out of the red blood cells, entering the bloodstream once again. At this point, malaria symptoms are evident. Some of the parasites differentiate into sexual gametocytes at this point. This is where the gametocyte phase starts. These gametocytes are taken up by another Anopheles mosquito during a blood meal, and the Plasmodium life cycle begins in the mosquito host. The parasite multiplies inside the mosquito mid-gut wall, and while in there, the microgametocytes fuse with macrogametocytes to form zygotes. The zygotes turn to oocytes that grow and rupture and release sporocytes. which made their way to the salivary glands of the mosquito to inoculate into the next human host. Plasmodium can also interact with environmental factors such as water, temperature, and humidity, which influence how it breathes, spreads, and affects health. Overall, Plasmodium and its interactions with other organisms and environmental factors are complex and dynamic. Its ability to avoid detection, persist, and spread within ecosystems. It's an important factor that has enabled its survival as one of the most dangerous human and animal parasites, affecting millions of people across the globe every year. In the year of 2021 alone, it was estimated that about 240 million cases of malaria happened. Most of these cases were presented in the African regions, affecting children under the age of 5. Plasmodium acts in a symbiotic relationship with the mosquito vector and the human host, benefiting from both. Pathologically, the Plasmodium has a detrimental effect on the health of the infected individual, causing symptoms such as fever, malaise, chills, vomit, anemia, jaundice, sometimes neurological complications, and even death, mainly also affecting infants and children under the age of 5. The environmental impact of Plasmodium is considerable, and it makes it difficult to mitigate. Populations of infected people and the associated mosquito vectors are usually confined to regions, leading to localized outbreaks. Additionally, due to the significant amount of resources that must be allocated to address the disease and its associated morbidity and mortality, governments in endemic regions are often forced into poverty. Also, due to the significant amount of insecticide used in order to reduce the population of Anopheles mosquitoes, runoff from application sites can be adversely affecting local ecosystems. In conclusion, Plasmodium malariae is a significant infectious disease with far-reaching implications for human populations, particularly those in tropical and subtropical areas. It can cause both symbiotic and pathological effects with possible dire health outcomes, and can also have environmental costs due to the resource expenditure, vector control, and vector killing runoff.
The HBV virus. The HBV virus stands for hepatitis B. It is an infection which causes inflammation of the liver, and it can be spread through blood, semen, or other bodily fluids from a person infected. The structure of hepatitis B is it contains an outer envelope and an inner core. The outer envelope is composed of a surface protein called the hepatitis B surface antigen, or HBAG, which stands for both acute or chronic. It can be detected by a simple blood test. As for the inner core of the virus, it is known as the protein shell. It is referred to as the HBV core antigen, which contains the HBV DNA and enzymes used in a viral replication. The genome of HBV is a partially double-stranded circular DNA of about 3.2 kilobase pairs. The viral polymerase is covalently attached to the 5 end of the minus strand. It is classified in 8 genomes and each have a distinct geographic distribution. And it also belongs to the Hypnaviridae family. It is known to be endemic to human population, but hyperendemic in many parts of the world, which basically stands for that it targets this population, but it's not just limited to humans. HBV induces oxidative stress and stimulates central carbon metabolism and nucleotide synthesis. Its unique features of the virus replication cycle present an ability to persist in affected cells. The life cycle of hepatitis B is it begins with 1, The virus enters the host liver cell, and is transported into the nucleus of the liver cell. Once inside the nucleus, the viral DNA is transformed into a covalently closed circular DNA, which serves as a template for viral replication. HBV virus can even survive outside the body for at least 7 days, and it can still contract someone once it enters the body who is not protected by the vaccine. Incubation period ranges for the virus from 30 to 80 days. Most adults tend to recover, but around 5 to 10% are unable to fully clear from the virus, and it then becomes chronic. Once the virus has contracted the host, the infected person can experience no symptoms at all to experiencing the following, which are fatigued, poor appetite, stomach pain, nausea, and jaundice.
Ebola virus. Ebola virus disease, also known as Ebola hemorrhagic fever, is a viral disease that can infect animals and humans. The virus is known for having a mortality rate between 55 to 60% for people who are infected. The forms of transmission are from animal to animal, spillover event, and human to human transmission. There is evidence stating that fruit bats are the reservoir host for the Ebola virus. There are 4 different Ebola viruses that fall into the genus Ebola virus, as seen below. Ebola virus was first discovered in 1976 when 2 people were diagnosed with hemorrhagic fever. The first case was reported in the Democratic Republic of Congo near the Ebola River. The second case was in South Sudan, and scientists first thought that the cases were regarding the same person. They then discovered that it was 2 different viruses from 2 different locations. Currently, there is only 1 vaccine and 2 different medications available for the Zaire Ebola virus, which is the most common of the Ebola viruses and has caused the largest and deadliest outbreak to date. Ebola virus is part of the Filoviridae family, and other viruses of that family are seen below, which all belong to the Monogenaviralis order. Ebola virus is filamentous and can form a 6 or a U shape. It is a single-stranded RNA that is protected by a nucleocapsid and a lipid envelope. The nucleocapsid is made of nucleoproteins, viral proteins 24, 30, and 35. The viral protein 40 helps with the virion structural integrity. Glycoproteins help the virus dock onto the cell's membrane, and the L polymerase enzyme helps with transcription and translation. The virion protein 35 also helps the L polymerase with replication. The process of infection comes in 7 different steps, the first being attachment. There are many ways that EBOV can attach to its host using attachment factors. Some of these factors can be human folate receptors or T-cell immunoglobulins. EBOV does not need specific receptors, so that is part of the reason why it can target various cell types. This attachment is also mediated by the glycoprotein spike proteins, which help the virion dock onto the cell's membrane. The next step is entry. Once the virus binds to a receptor, it can enter the cell in 3 ways. The most common form of entry is macropinocytosis. The next step is when EBOV has entered the cell and its 2 glycoprotein subunits, GP1 and GP2, are separated. GP1 is broken from proteins into amino acids by the capsin B, L, and thermolysin. This breakdown then allows the next step to occur, which is when the virus interacts with the cholesterol transporter Niemann-Pick C1, which is important for entry into the cytoplasm. Once into the cytoplasm, the last 3 steps occur, being transcription, translation, and exiting. This is catalyzed by the L polymerase enzyme. An antigenome is also created and used to make progeny genomes. These progeny genomes can then be used for more transcription and translation, which are then transported to the surface of the cell's membrane. It is then released into the extracellular space via budding. During this budding process, that is where the virion gets its lipid envelope from. Ebola reproduces via the lytic cycle since it uses the host mechanism for transcription and translation to reproduce itself once it enters the cell. EBOV has many ways to fight against the body's innate and adaptive immune system. In fact, monocytes/macrophages and dendritic cells were found to be the first types of cells that are infected with EBOV once entered into the host, most likely due to the fact that they are major components of the immune system and help with responses to pathogens. It attacks the innate immune system by preventing dendritic cell maturation, which impairs their performance and ability to fight off viruses. It is also able to produce massive amounts of cytokines, which can lead to organ failure and even death. EBOV also alters the adaptive immune response by inhibiting the antibodies produced by the B cells, which are responsible for tagging pathogens for destruction. One study also found that EBOV can affect our environment, still being viable 14 days after its release from the body in a climate-controlled hospital conditions, and being viable after 4 days in tropical conditions. This information is very important from a pathological and symbiotic standpoint because it shows that the virus can stay viable days after its release from the body, something healthcare workers need to keep in mind when caring for an infected individual. All items that come into contact with the virus need to be disposed of correctly, or else it can cause a healthy individual to be infected. That person can then go on to affect another person, thus causing an increase in the rate of infection.
Today I'm going to talk about one of the most feared microbes, herpes, also known as human herpes virus 1. But first, let's dispel a little stigma surrounding this very smart virus.
Subby says that you were freaking out because you think you have herpes. What the Seriously, relax. People are way too full of shame about herpes. You told her?
Ux has herpes too.
Yeah, along with many, many of the coolest people on Earth. And I'm only telling you because I want to mitigate any unnecessary suffering that comes from the stigma surrounding a skin blemish. Treat it the best you can, tell your partners, and if it grosses them out, they're losers. Move on.
Yeah, okay, none of your business. Yes, actually, thank you.
There are 8 different kinds of herpes viruses that affect humans, and they're numbered in the order in which they were discovered. Previously, HHV1 and HHV2 were formerly known as HSV1 and HSV2, but the name has since been updated. The most common collectively infect about 90% of the US population, and they present themselves as oral and ocular herpes, genital herpes, chickenpox and shingles, and mononucleosis. All viruses in the family Herpesviridae have 4 main anatomical components: an envelope with glycoprotein spikes, a tegument with proteins, and a capsid that protects the DNA. The envelope is the outermost surface and is composed of a lipid bilayer with multiple copies of more than 12 viral glycoproteins that enable recognition, attachment, and entry to the host cell. The space between the envelope and the capsid is called the tegument and contains more than 20 viral proteins, which enter the cell at initial infection and play a role in the transcription and translation of viral genes and shutting down host antiviral defense mechanisms. The capsid is an icosahedron shell structure that encloses and protects the genome. The DNA of HHV1 is linear, double-stranded DNA with 152 kilobase pairs long. As a virus, HHV1 does not have the ability to metabolize on its own. It is an obligate intracellular parasite, which means that it will take over the host cell's machinery to produce new virus particles for replication. In the process, it will cause harm to the host, but will not kill it in order to preserve its own survival. The process of replication begins with attachment. The surface glycoproteins of HHV1 will tether to the host cell membrane receptors. This binding triggers other glycoproteins on HHV1 to execute membrane fusion. The glycoprotein will insert into the host cell and then refold, This repeated action happening over and over again brings the host cell and the virion together while creating a pore in the host cell membrane. Then the nucleocapsid containing the DNA will enter the cytoplasm through this pore. Once inside, the capsid will travel to the nuclear membrane. As it's traveling, the tegument protein surrounding it will detach from the capsid and move throughout the host cell to perform functions that assist with cell machinery takeover. The capsid will bind to the nucleus and release the viral DNA through a nuclear pore. Then a series of transcription and translation occurs both inside and outside of the nucleus to generate genes and proteins that ultimately form vironucleocapsids inside the nucleus. From there, it will bud through the nucleus, taking on a portion of the membrane for its own. It moves through the endoplasmic reticulum where it will pick up glycoprotein spikes, and then it passes through the Golgi apparatus before exiting the host cell membrane. After lytic replication in epithelial cells, the virus will establish latency by reseeding into the ganglia of nerves. There, it will deposit the viral DNA into the nerve cell nucleus, where it will then hide out. And during this time of latency, the virus will be relatively inactive. But it can be reactivated by triggers such as a fever, emotional stress, hormone imbalance, UV exposure, trauma, and immunosuppression. HHV1 most commonly establishes latency in the trigeminal ganglia. This means that it will most often travel down the mandibular nerve, resulting in cold sores and oral herpes around the mouth. If it travels down the ophthalmic nerve, then it results in ocular herpes. If the virus establishes latency in the brachia ganglia, then it will travel down the arm and cause a finger lesion called the whitlow. It is estimated that this virus affects 3.7 billion people. It results in lesions on and around the lips, gums, eyes, and fingers. It can be as rare as having one outbreak every 4 years to every 2 weeks. Many people don't even know that they have HHV1 or HHV2 because signs and symptoms do not present in everyone. In very rare cases, people with severe immune suppression, such as HIV, can get encephalitis and meningitis if the virus travels to areas of the brain. New research is even suggesting that because of this, there might be a link between HHV1 and Alzheimer's disease. HHV1 only affects other humans through skin-to-skin contact, and there must be damage caused to the mucosa or the top layers of the skin in the new host. These can be caused by microscopic ruptures that can occur by abrasion or intense rubbing. The virus will enter the epithelial cells or the mucosa of the new host and produces inflammation and cell death. Sometimes this is also accompanied by malaise, fever, and muscle pain. But let's end on a good note. HHV1 may provide symbiotic impact on the host over time. The innate immune system knows HHV1 is present, even though it is latent. So this presence essentially trains the innate immune system to be on high alert, constantly scanning for pathogens. It can't remember specific pathogens the way that the adaptive immunity can, but it remembers that they exist. A 2007 study showed that HHV1 mice were resistant to both the flag and serious foodborne infections.
I completed this project on one of my least favorite viruses, the human papillomavirus. HPV has been around humankind for a very long time, as the first description of HPV symptoms was in 4th century BC. Scientific investigations in the mid and late 20th century gathered further information. HPV is incredibly prevalent around the world, as almost everyone who is sexually active will be infected with HPV at some point in their lifetime. However, the majority of infections are destroyed by the immune system within 2 years, and most people with HPV show no signs or symptoms. HPV strains are divided into high and low risk. Low-risk HPV strains can cause warts, such as those found on genitalia or in the oral cavity. High-risk HPV infections can cause cancer at the site of infection, including cervical, anal, oropharyngeal, penile, vaginal, or vulvar cancers. For each of these different types of cancers, HPV is the leading cause. Globally, more than 600,000 people are diagnosed with an HPV-associated cancer each year. In the United States, HPV has an unequal impact on racial and ethnic groups with increased mortality and incidence rates of cancer among people who are Hispanic, Black, and American Indian or Alaskan Natives. These inequalities are likely the result of an interplay of several factors, including unequal access to healthcare, bias against minorities and historically excluded communities in the healthcare system, socioeconomic status, and individual level factors. Globally, low and middle income countries experience the majority of deaths from HPV-associated cervical cancer, a result of global inequalities in access to the HPV vaccine and comprehensive medical care, prevalence of HPV, gender discrimination, and poverty. At a molecular level, HPV has a double-stranded DNA molecule, is naked, so without an envelope, and has an icosahedral capsid. The DNA is circular and includes histone proteins and 72 capsomeres that are joined to create the capsid structure. HPV is a relatively small virus, only 52 to 55 nanometers in diameter, with about 8,000 nucleotide base pairs within its DNA. There are more than 180 strains of papillomavirus that infect humans, and there are 5 genera, including alpha, beta, gamma, mu, and nu papillomaviruses, and they are a member of the Papillomaviridae family. Papillomaviruses do not just infect humans, but also infect many others, including dogs, cows, European elk, deer, reindeer, sheep, horses, birds, rodents, rabbits, cats, porpoises, hamsters, and possums. All of the species of papillomaviruses are divided among 53 genera and 2 subfamilies. Papillomaviruses are close in relationship with polyomavirus, and previously papillomaviruses and polyomaviruses were grouped into the same family, given their similar shape, anatomy, DNA structure, and that both can cause cancer. However, differences between the two include that polyomaviruses are connected to Merkel cell carcinoma, prostate cancer, and colorectal cancer. Transmission of HPV is either skin to skin or skin to mucous membranes, most commonly through sexual activity. However, other forms of transmission include via fomites, non-sexual contact, or from birthing parent to child, such as through amniotic fluid or the placenta, and during childbirth. After whichever transmission route is taken, HPV enters through a microabrasion in the stratified squamous epithelium and targets basal cells at the bottom layer. HPV then uses a capsid protein to attach to receptors on basal cells or on the basement membrane and is able to gain access to the host cell via endocytosis. From there, HPV moves through the cell to ultimately complete its journey at the nucleus. Here, HPV penetrates into the nucleus through nuclear pores or by taking advantage of the normal cellular process of mitosis and the required dissolution of the nuclear membrane. HPV sheds its capsid after entering the nucleus and finds promyelocytic leukemia or PML nuclear bodies to begin its own transcription. As seen in the painting here, this is a magnified version of the HPV life cycle. Transcription takes place and the HPV genome is, at first, replicated into no more than 100 copies within each nucleus. To evade the host's immune response systems, the viral proteins do not accumulate in large numbers, thereby allowing viral replication to take place undetected. Also, uniquely, as HPV targets the basal stem cells, it exploits the host cell differentiation process. During the early and mid stages of viral replication, HPV utilizes some of its proteins to suppress cell apoptosis, as well as block cytokine release. In the final stages of viral replication, a late promoter sequence is turned on and many of the viral proteins are amplified, now creating thousands of viral genomes and capsid proteins, which assemble to create the icosahedral capsid. As seen in the green cells, the virus matures in keratinocytes, present on the top layer of the epithelium. HPV is shed from here and the free virions are able to infect nearby cells. HPV also interacts with regional microbiota. Vaginal microbiota is normally characterized by an abundance of lactobacillus species, which causes a relatively limited diversity of microorganisms present, but also helps to lower the pH and prevent pathogen invasion. Individuals with HPV have higher levels of all of these bacteria listed, and it appears that HPV alters microbiota, generating greater diversity in microorganisms present compared to those without the virus. 3 other bacteria that are not listed are all associated with the removal of HPV from the human body. The HPV vaccine was first developed in 2006, and since then more vaccines have been developed, all of which utilize an HPV protein, so no RNA or DNA, to effectuate an adaptive immune response to this specific virus. It's recommended for people between ages of 9 and 26 and is given in a 2 or 3 dose series. The vaccine is effective at preventing genital warts as well as the 6 different types of cancers. It is imperative to underscore that the development of this vaccine would not be possible without Henrietta Lacks, a Baltimore mother who, in 1951, was diagnosed with cervical cancer at Johns Hopkins. Prior to her passing, Ms. Lacks' cancer cells were removed and sampled for research, quote, without her knowledge or consent. The HeLa cells were considered immortal and the first to grow without issue in a research lab. The HeLa cells were then shared globally and had a profound effect on medical advancements, all without her surviving family's knowledge for more than 20 years. To date, the HeLa cells have been material to 75,000 scientific inquiries, including HPV research. Ms. Lack's immortal cells directly led to the finding of a connection between HPV strains and cervical cancer and to the HPV vaccine's development.
Rhinovirus, also known as the common cold, is the most common viral infectious agent in humans. First discovered in 1956, rhinovirus is responsible for more than 1/2 of cold-like illnesses and costs billions of dollars annually in medical visits and missed days of work. The human rhinovirus, or HRV, has an icosahedral structure, meaning it is a spherical geometric shape composed of 20 sides. These 20 sides are made of equilateral triangles composed of 4 proteins, V1, V2, V3, and V4. Rhinovirus is a non-enveloped, positive-sense, single-stranded RNA virus. The outer surface of the virus has 60 spike-like protrusions that act as receptors and bind to host cells. Rhinovirus is also one of the smallest types of virus, with a diameter of around 30 nanometers. Most other viruses are around 10 times this size, with diameters of about 300 nanometers. Rhinovirus belongs to the picornavirus family and also belongs to the enterovirus genus. The picornavirus family causes a wider range of illnesses than almost all virus families. Their ability to mutate faster than DNA or double-stranded viruses make them easy to contract and difficult to prevent. Contracting rhinovirus is as simple as coming into contact with someone who currently has the virus or a fomite that is contaminated. The virus spreads when an individual coughs or sneezes and water droplets are released into the air. Once these droplets come in contact with another host, the cycle continues. HRV is most likely to enter a host through the nasal cavity. Finger-like projections on the outside of the virus bind themselves to host cell receptors. Once attached to the host receptor, penetration and uncoating occur. The virus is able to inject its RNA into the host cell, which will directly translate the viral genetic material into many more infectious progeny. In response to this viral replication, inflammatory cells become activated. These symptoms include sore throat, sneezing, coughing, body aches, nasal congestion, and fever. There are many over-the-counter antiviral products that help to battle the symptoms of rhinovirus. However, these medications are not a cure. And since there are over 160 strains of rhinovirus, it is nearly impossible to develop a vaccine that will prevent HRV. Until then, keep washing your hands.
Poliovirus is the viral pathogen that causes poliomyelitis, a debilitating disease that can cause paralysis when nervous tissue is infected. Poliovirus was first identified in a lab by Karl Landsteiner and Erwin Popper in 1908. Their research inspired others who were able to produce the first vaccine for poliovirus decades after their discovery, the first in the 1950s. The first person to give us an image of poliovirus was the esteemed scientist Rosalind Franklin, who used X-ray crystallography to give us the initial image of this paralyzing virus icosahedral shape. Poliovirus is a type of enterovirus belonging to the family Picornaviridae, which are a family of positive-sense single-strand RNA viruses that have a non-enveloped capsid in an icosahedral shape. Viruses in the family Picornaviridae have a relatively short nucleotide sequence in their RNA, roughly 7,500 nucleotides in length. Other genera in this family include the hepatovirus, which causes hepatitis A through E, and rhinovirus, the cause of the common cold. As it is a virus, poliovirus does not attain or use nutrition, does not grow, and does not have its own metabolic system, instead relying on the nutrition, growth, and metabolism of the cells that it infects to replicate itself and infect further. Like other viruses, poliovirus is inactive outside of a host cell and does not actively seek out cells to infect since it doesn't have the complexity to do so. Instead, it relies on the random occurrence of bumping into a host cell that also happens to have a certain receptor, specifically the aptly named poliovirus receptor, CD155, which is only found on human cells. Humans are the only known natural host of poliovirus, and human epithelial cells produce high levels of CD155 RNA, suggesting these cells might be primary sites of poliovirus replication, meaning the oropharyngeal and intestinal mucosa are particularly adept cells for poliovirus replication. Being non-enveloped, poliovirus cannot attach to host cells using membrane fusion and instead relies on direct penetration where the viral capsid attaches to its host cell via membrane receptor CD155, sinks into the cytoplasmic membrane via a pore, and only the viral genome enters the cytoplasm. Within the cytoplasm, poliovirus can replicate its genome extremely easily, as it is a positive-sense single-strand RNA virus. Positive-sense single-strand RNA is very similar to mRNA, which gets directly translated into proteins by the ribosomal complex. Once the viral genome and capsid proteins are translated, assembly of new virions occurs within the cytoplasm spontaneously and the new virions are expelled via exocytosis, lysing the host cell. This process occurs about 4 to 6 hours after the poliovirus first infects the cell, and for every host cell infected roughly 10,000 new virions are released. Our immune system is extremely effective at responding to infective agents, including viruses. We are all born with innate defenses that respond almost immediately via chemical and hormonal signals at the first sign of injury or infection. However, poliovirus has a few tricks for evading our body's defenses. Cytomegalovirus is a relatively common virus in the herpes virus family that remains latent in the body for life. The cytomegalovirus contains UL141 protein that blocks surface expression of CD155, leading to evasion of NK cell-mediated killing, and therefore makes it easier for poliovirus to evade the immune defenses that would normally tag it as non-self and destroy it. Poliovirus and its relatives in the Coxsackie viruses can avert immune antibody action, despite the presence of possibly high titers of neutralizing antibodies and cross-reactive T-cell help against the first serotype, meaning that a person who was infected by one strain of poliovirus is able to be reinfected repeatedly by different serotypes. It is difficult to comprehend the fear that infection with poliovirus once wreaked upon our population. In modern times, we rarely, if ever, hear of anyone being afflicted with poliomyelitis, the paralytic, nervous system damaging form of infection that poliovirus causes in roughly 1% of cases. In mild infection, symptoms are innocuous and barely discernible. Fever, malaise, sore throat, and nausea are hardly even regarded, as they could be from any common illness. Nervous system infection symptoms are much more sinister, starting with neck stiffness, photosensitivity, and a pins and needles feeling in the limbs and eventually leading to difficulty breathing or swallowing, and possible total and permanent paralysis of the body. There is evidence that if infection were to occur in a person whose tonsils or adenoids were recently removed, they would be far more likely to contract the paralytic form of polio since nerves are exposed at those sites. There are many ways to spread poliovirus, including not washing the hands after using the bathroom or changing a diaper, drinking contaminated water or getting it in your mouth, eating foods that have touched contaminated water, swimming in contaminated water, coughing or sneezing, being in close contact with an infected person, or touching contaminated surfaces. Infected individuals can shed virus in the feces for up to 35 days, even after symptoms have subsided. The advent of modern sanitation practices and a worldwide vaccination initiative starting in the 1950s has severely limited the range of poliovirus infection. Due to these efforts, 2 of the 3 serotypes of poliovirus have been eradicated, and the final, 3rd serotype is only found in 2 countries of the world, Pakistan and Afghanistan. The oral polio vaccine, Replicate-S, in the intestinal mucosa and lymphoid cells in the oropharynx and intestine, in a similar manner to wild poliovirus infection, which means that there is potential for a person to become infected with the virus if they come into contact with the vaccinated person's fecal matter for a certain period after vaccination. However, this risk is not present with the inactivated vaccine. It is very important that all people who are capable of receiving vaccines get them, for they protect everyone from dangerous diseases like polio.
In the world of neurological disorders, there are very few that are as rare and mysterious as transmissible spongiform encephalopathies, also known as prion diseases. Prion diseases are found in both humans and animals, with some prion diseases that are able to be passed from infected animals to humans. These diseases are fatal 100% of the time, and there are no cures or treatments to slow the progression once the proteins in the brain are infected. So what are they? Prions are naturally occurring proteins found in all of us. They're known as PRP, C, or cellular prion proteins, and they are mostly located on the surface of cells within our central nervous system, and it is unknown what their exact function or purpose is. The transformation of these proteins happens when PRPC is infected with the isoform PRPSC, or scrapie prion protein. This infection leads to a profound alteration in the PRPC protein structure, causing it to become folded, misshapen, and pathogenic. Once the PRPC protein is infected with PRPSC, the infection cascades almost like dominoes. Any protein that is near the PRPSC protein then also becomes infected with PRPSC, and so on and so forth, until there are so many infected proteins they cannot function. Once there are so many of these proteins unable to function, they clump together and the brain triggers an enzyme response to try and break them down. Unfortunately, PRPSC proteins are resistant to that, and the continual clumping and replication of these proteins eventually causes holes in the brain. So what do they look like? PRPC protein is a monomer consisting of 2 distinct domains, the unstructured N-terminal and the structured C-terminal. The C-terminal is globular and characterized by 3 alpha helices and a short anti-parallel beta sheet. The entire protein is composed of 253 amino acids. Once the PRPC protein is infected with PRPSC, the structure changes dramatically. The alpha helical structure decreases by about 30%, and the beta sheet structure increases from about 3% all the way up to about 43%. This extreme change allows for the infected proteins to become prone to aggregation, which is when the misshapen protein produces amyloid fibrils, which end up stacking up on top of each other, causing the clumping I was talking about earlier. So I'll show you my artwork now. So I drew a brain with holes in it, and then this is a representation of a healthy protein. So these are the helical alpha helical structures, and then these twisty ones are the beta proteins. And then down here, I did a representation of an infected one. And so the helical structure, the green ones are now straight up and down, and the pink and the blue have straightened out and flattened out. So yeah, there's that. One of the reasons these proteins are so hard to study and understand is because once they become infected, the amyloid fibrils can exist in multiple different structural forms, even though these proteins have the same sequences. There's no rhyme or reason that has been discovered yet to tell us why this happens. Once a protein is infected, it replicates rapidly and similarly to the way viruses do, and the main difference is that these proteins do not possess any genetic material. The most common human prion disease is called Creutzfeldt-Jakob disease, and it manifests in 3 major forms: sporadic, hereditary, and acquired. Sporadic is the most common, which makes up about 85% of infections, and it's called sporadic CJD because there's no known cause for the protein infection. The symptoms mimic other common neurological disorders like Huntington's disease and Parkinson's. Initial symptoms include lack of coordination, memory loss, judgment issues, Behavior changes, confusion, insomnia, and vision changes. Once the disease progresses, symptoms intensify and can lead to blindness, speech loss, and inability to move. And ultimately, patients with CJD typically fall into a coma and die, and usually rather quickly, within 1 year of symptom onset. And while the end stages of this disease are believed to be painless, the myoclonic seizures and disorientation experienced by patients can be really distressing for their caregivers. And since there's no current cure or effective treatment to slow the disease progression, all care is palliative and focused on relieving their symptoms and providing comfort. Neurologist Hans Gerhard Creutzfeldt and Alphonse Marie Jacob described the disease in 1920, but they had no idea what it was, and neither did anybody in the scientific community. And then in 1938, Coulier and Schell described and found what they called scrapie, which was found in sheep. And because it had a long incubation period, it was theorized as a slow virus. And then in the 1950s, another disease called Cruru, which is also a prion disease, was discovered and presented with so many similarities to CJD and scrapie that scientists really were pushed to try and figure out what this new, quote, viral infection was. So scientists took the scrapie agent and infected mice and primates with it, and then tried to eradicate that scrapie agent from those animals using common antiviral methods. And it didn't work because prion disease is not viral, and all of the animals ended up dying. And then in 1966, there's this really badass woman named Tikva Appler who was doing research on the scrapie agent, and she presented a hypothesis that the scrapie agent might be of protein origin, and she was correct. It was not an accepted theory or hypothesis in 1966 because it directly challenged the central dogma of biology. But then in the 1980s, the same protein only hypothesis was refined and represented by Weissman and was widely accepted in the scientific community as the main hypothesis for prion diseases. And it has really been a jumping off point for much of the research into other neurodegenerative disorders. Thanks for listening. Bye.